So let us see. Let us try to understand the division of last lecture. So that is DNA replication, right? So what are the major steps in DNA replication? DNA replication has 5 steps in my knowledge. Am I correct? Yeah. What are the 5 steps we have? Initiation. Yeah, one second. Initiation. Initiation. Then primer synthesis. Elongation of primer. Then primer removal and gap filling, and termination of replication. So in the initiation we have 2 important steps. What are those? Yeah, in the initiation we have origin of replication plus unbinding of DNA. Unbinding of DNA is mediated by helicase. Then helicase unbinded, then what happened? Yeah. So maybe we will draw a diagram along with the points, right? So first we have a DNA. So this DNA was unbinded here. So unbinding of DNA happened. So this is the unbinding portion. So maybe this is still like this. So here the DNA was unbinded. DNA unbinded. So this unbinded portion is called as replication fork. This area is called as replication fork. Now we have 5' to 3' and 3' to 5'. And our helicase only, this one. Polymerase only works from 5' to 3'. And the direction of replication is this side. Right? So first we have the origin. From origin we got unbinding of DNA. Unbinding of DNA is facilitated by. So I'm giving here unbinding of this enzyme is helicase. So helicase unbinded the DNA. After unbinding, what happened next? Now SSB proteins will coat here. So they will maintain that unbinding status of the DNA. SSB proteins will maintain the unbinded portion and it will prevent the supercoiling. There is one more supercoiling prevention thing. So these are called as SSB proteins. SSB proteins are kept in their place, right? After SSB proteins, what happened? So topoisomerase. There is one more enzyme, topoisomerase. So what is the role of topoisomerase? I am giving topoisomerase here. So topoisomerase it will prevent supercoiling of unbinded DNA. Unbinded DNA supercoiling will be prevented by topoisomerase. So far clear, right? Yeah. These are the major steps in initiation. Then what is the next one? Primer synthesis. Then now a primer will be binded to the 5' to 3' end, right? So a primer will be binded. So this binding of primer is called as annealing. Primer binding is called as annealing. Annealing means here the primer was binded. Primer binding is called as annealing. Done. The primer was binded now. What is the meaning? Annealing. Okay. Yeah. Now this primer will act as a starting point for the replication. Starting point for the elongation of the DNA. Right? Replication of the DNA. Next one. Next one is elongation. Now the primer will be elongated. Okay. So elongation. So in elongation, what is the major enzyme we are getting here now? Now we got the DNA polymerase. So in elongation, the first one is DNA polymerase. So please don't talk. Okay. disturbing the lecture. Then this DNA polymerase also binded with the major strand by a clamp. What is that clamp is called as? Sliding clamp. So I am just writing here sliding clamp. It will be binded to the DNA polymerase. Then what is the role of DNA polymerase? Now this DNA polymerase, similarly here also same thing happens parallel. One more DNA polymerase, sliding clamp and primer. The problem is, since the overall replication going this way, doing this strand was very easy. But this strand will be very tough because behind it, it's already going that way. Behind the DNA, it's unwinding that direction, but it is unable to cope up with it because it's going reverse direction. When the train going that direction, we are moving against it. So definitely our movement will be slowed down. The person who is going that side will be faster in action compared to us. Similarly, here this strand is called as a lagging strand. Lagging strand will be formed, and here this strand is called as leading strand. So leading strand was faster in action, whereas lagging strand is slower. That's what it's called as. Leading strand and lagging strand. Now, due to this lagging strand, when the DNA is not moving this side, there will be a small fragments left in between this polymerase activity. Those fragments are called as, so yeah, lagging, I'm writing the lagging and leading. Then we have the Okazaki fragments. Now this Okazaki fragment gaps will be filled by one more enzyme. What is that enzyme? That we'll see later. Helicase, we'll see later. So these are the steps, major steps in the elongation of the primer. Okay, primer elongation is facilitated by polymerase, connect with a clamp, and we have a leading strand and a lagging strand preparing Okazaki fragments. So clear. Next one. Next one is primer removal and gap filling. Now we need to remove this primer and we need to fill the gap again. So yes, right now. So RNase 1. Now there is an enzyme. This enzyme is called as RNase. RNase 1. It will remove the primer. RNase 2. Oh, sorry. RNase 2 will remove the primer. RNase 2 remove the primer. After removal of primer, what happened? Now polymerase 1. See, this primer removed, and now there is a gap. Maybe I will keep gap here. Now there is a gap in between primer removal. Now the gap will be filled by one more enzyme. We have one more enzyme that will fill the gap of the primer removed side. That enzyme is called as polymerase 1. DNA polymerase 1 will fill the gap. DNA polymerase 1 will fill the primer gap. Primer gap is filled by DNA polymerase 1. Is it clear? Yeah. After filling the gap, last and final is, yeah, these Okazaki fragments. Fragments are, a pieces of strands of those fragments has been formed. In between those fragments there is a gap, and the gap should be filled. So Okazaki fragments gaps will be filled by DNA ligase. There is one more DNA enzyme that is filling these Okazaki gaps. Okazaki gaps are filled by DNA ligase. Ligase activity is always binding. Okay, it will connect something. So this is the overall picture of DNA replication in a nutshell. Clear? So yeah, so ligase, DNA ligase. Then I haven't discussed this previously. Listen, see students, for every DNA, the portion of the DNA that codes a protein is called as, the portion of DNA that codes a protein is called as what? The portion of DNA that can able to code a protein. That portion of DNA is called as genes, right? So what is the difference between a gene and a DNA? How would we differentiate or how would we recognize the gene sequence from the DNA sequence? Because DNA also have ATGC base pairs, even gene also has ATGC base pairs. So how to recognize a gene? So I haven't discussed previously, every gene has its starting codons and ending codons, promoter regions and terminating regions. Okay, so there are 5 to 6, we can say like this, in Harry Potter he will use some magical words, right? So similarly there are 5 to 6 magical sequences that indicates the starting command. So if there is AATT, that indicates starting of the gene. And termination also, it has a particular sequences, 2 to 3 types of them. I don't know, I need to check again. So if you reach those TTTTU might be a termination codon. So promoter codon to termination, this might be a gene. So then obviously the gene unwinding happens near a promoter region, not randomly. The gene will be unwinded, the DNA will be unwinded at the promoter region. And it will be ended at the termination happens at the terminator region. So there are certain base pairs that will code the starting and the ending of the gene. Okay, that we will see. The last one is termination of the replication. So once you got the termination sequence, it will stop the elongation of the primer and it will terminate. Now after terminating, I have said you that if you see the structure of a chromosome, what does it has? It has a short arm, it has a long arm, P arm and Q arm, and there is a centromere at the center. This is the basic structure of a chromosome. At the end of the chromosome, we have what? The end of the chromosome is called as telomeres. Telomeres. Now, upon terminating of replication, it will refill the telomers, only present in eukaryotes. In eukaryotes, we have this mechanism right now, that is telomer replication. Telomer replication. Telomer is just a repeated sequence of any one of the base pair, ATT, AATG, any one of the base pair. I don't know much, but there is an exact sequence for the telomer. So telomer replication happens. What is the significance of telomer replication? Why do we need telomer need to be present? To preserve the DNA, it will determine the age of the cell, the number of the cell divisions which happened previously. So it's a record of the number of cell cycles.
Preserve DNA.
Preserve the DNA and the age of the cell. It will determine the age of the cell or the number of previous cell divisions happened to it. Preserving the DNA determines the age of the cell, and also it will determine the previous cell divisions, number of previous cell divisions, how many times this cell has underwent the division. I will tell you one more thing, students. At one particular point, the telomere length will be very small, and if the telomere was very short, there is a gene that indicates P53 gene. I am telling you a very simplest way. Once this P53 gene got exposed at the end of the chromosome, immediately this P53 will give a signal to the cell that now you should die. The cell will be committed suicide, because if we are entertaining this cell might become a mutant and it might become a cancer. So there is a programmed cell death. Once the telomere gone, the P53 will give a signal to underwent a senescence. Senescence means the cell won't divide further. That is called as programmed cell death, or it is also called as apoptosis. Apoptosis signal will be given by the cell. Yes. Ashley. Program cell. Program cells death or apoptosis is said. Once the telomere gone, it will undergo apoptosis. So they will destroy. So the body has an inbuilt mechanism of maintain the new generation of new cells. By the way, who are responsible for maintaining new cells? They are actually stem cells. Stem cells will undergo differentiation and they will replace the old cells. Okay. That will be very rare, but just keep in mind. Then last and final, fourth convergence present in prokaryotes. Fourth convergence. The last step is fourth convergence. Fourth convergence is, again, this replication fork will be rewinded back to its original position. Okay, this is called as fourth convergence. This fourth convergence is mediated by, I didn't give the example of fourth convergence. So there might be an enzyme that is responsible for the fourth convergence. Okay, so that's it. This is all about the DNA replication and what type of replication is this? The type of replication happens in humans. There are three types of DNA replications. What are those? Conservative, semi-conservative, and dispersive. So in humans, what type of replication happening? Semi-conservative. So what is the what's going on in semi-conservative mode of replication? What happens? There is one parent strand and there is one new strand. So that is called a semi-conservative replication. Very good. Then write down further notes. I have discussed previously. DNA also have a replication also have a proofreading mechanism. Proofreading mechanism. What do you mean by proofreading mechanism? See, obviously A must bind with P, G must bind with C, right? GC. If unfortunately A binded with G, they will identify such errors and these errors will be rectified. If we are not rectifying these errors, it will become a mutant and the cell cannot function properly. I hope you can understand the consequences of mutations. It might become a cancer cell. So, proofreading by DNA polymerase. So, write down in this, proofreading can be done by two methods. One, with the help of DNA polymerase, proofreading mechanism. Proofreading is done by two methods, with the help of DNA polymerase, and other one with the help of mismatch repair mechanism. Mismatch repair mechanism. I said that polymerase will always work from 5' to 3'. Am I correct? Also, parallelly, the same polymerase also checking 3' to 5'. See, polymerase enzyme usually elongate the primer from 5' to 3'. Same polymerase, what it was doing is, it was also proofreading from 3' to 5'. It is also proofreading 3' to 5'. That means here it is synthesizing and here it is also proofreading, whether the bottom was properly aligning or not.
From bottom to 5' to 3'.
5' to 3' to 5'. So at the bottom it is also doing proofreading mechanism from 3' to 5'. When it is moving like this, here it is elongating the RNA and here it is proof checking the complementary base pairs. So from 3' to 5', it is also checking an activity called exonuclease activity to remove incorrect bases. At the bottom, it is doing exonuclease. Exonuclease means removal of nucleic bases, the abnormal nucleic bases from the bottom. It is doing exonuclease activity and remove incorrect base pairs. Incorrect base pairs are removed by the polymerase enzyme. This is one proofreading mechanism. Okay, polymerase not only elongating the primer but also checking the mismatched base pairs at the 3' to 5' bottom. That is complementary structure. And next one, mismatch mechanism. So it will identify any mismatch and it will be replaced by appropriate base pairs. If A, it should be always with T. If it is G, it will remove the G and it will replace the T. Got it? Yeah, that is called as the error correction mechanisms or proofreading mechanisms. And I said previously, new students you came today, all these things happening at a rate of 2000 replications per second in a single cell. If you see the hemoglobin, not 2000, 20,000. A red blood cell can able to prepare 20,000 hemoglobin molecules every second. You just imagine how fast these things are working in the body. Too fast actually. Enzymes are so fast. Yeah. So I think I need not to say much about significance of DNA replication. What is the significance of DNA replication? So the significance is genetic continuity to maintain to continue the genes in the future, the generations. Then growth and development of the body. The current growth should be facilitated by DNA replication. Write down significance of DNA replication. Significance includes genetic continuity. Second, growth and development. Third one, repair mechanisms. It also repairing the problems, repair mechanisms, and also genetic continuity, then growth and development, and third one, repair mechanism. This class is until 11, 10. Okay, I have got it. Repair mechanism, and fourth, it is the basics of hereditary. It plays as a basic foundation for hereditary. Hereditary means you know that. Take it from fathers to take it to the next generation. It is the foundation for hereditary. Very good. Can you able to tell me the difference between applications and significance? What is the meaning of applications? Applications of replication. Can you tell? In what areas we can apply? Application means the uses in multiple areas. What might be the applications of DNA replication? Yes? Genetic modification. Oh, so you want to make some hybrid animals, hybrid organisms. Very good. To make any, those things are called as hybridization. For hybridization, very good. Hybridization. I hope you understood what is hybrids, right? We have a bad tomato plant and we have a good tomato. We hybrid. We grafted them. Genes are shuffled. Very good. Hybridization to make a GMOs. Can I make GMO? Genetically modified organisms can be created. Very good. Any more? Any other applications? Forensic analysis? Forensic studies? Are there? Study. What studies?
Like reading about what's happening,
Research. You are doing a research to understand how it's working. For a research purpose, for forensic analysis, for hybridization of GMOs. Anyone else? Actually, you can give many examples for DNA. Any more applications? Molecular diagnosis. Excellent. Molecular diagnosis. I know that there is a woman, very great achievement. In the women they are getting breast cancer, and if we identify exactly what gene mutation causing this breast cancer, this aggressive growth of this mammalian tissue, okay? If we identify that gene, we can literally target the gene by a drug. Just suppress the gene and you won't get a breast cancer. And one scientist identified that, and it took 20 years for her to pinpoint the exact gene responsible for this breast cancer. And she got Nobel Prize. And she has saved millions of people. Today we can easily, there is a marker for BRCA gene, BRCA. That gene responsible for breast cancer. If we identify this gene in the early days, there is a chance of survival. We can easily treat this breast cancer. So yeah, obviously that is for molecular diagnosis. Identify the molecular marker, treat it with the medication. Molecular diagnosis, very good. Anything else? Biotechnological examples. Biotechnological applications. Production of insulin. production of extraction of proteins, biotechnology. I mean, not to say, yes. Tissue repair. Tissue repair. Of course, transplantation. Organ transplantation. We need to understand the HLA typing, the matching of the donor and the graft. Matching of the graft to donor and recipient. Very good. Any more examples? Tissue repairs. You know, they are saying that the advanced research was we can even prolong the human longevity, age. We can live longer life if we are able to contain the damage of telomeres, because it is the telomeres that are determining the age of the cell. You try to protect the telomere length. The greater the telomere, the better the angle. The better we will live, the longer the life. See, there are some super animals, I can say, they used to live long times. Recently, 6 months ago, I saw in shorts, the oldest living tortoise or turtle, 203 years old. It lived 200 years. You just imagine, 6 to 8 generations of humans. My son, his daughter, very weird. How is that possible? Is it possible for us to, hybridize tortoise genes to us? Yes, starfishes can regenerate, and there are some organisms, some animals called exons. It's a type of salamander fish that if you cut the hand of the salamander fish, it will regrow. How is that possible? Growing an entire limb, something really miracle in the biology. So yeah, octopus also limbs will be regenerated. Yeah, such wonders are there. So we are doing that research to expand the longevity of the humans for the sake of colonization in the future. We want to live in another planet, so try these things. Let it be. Anyway, so next one there is a small topic called Okazaki fragments. I hope you know what are Okazaki fragments. Okazaki fragments are formed due to an outcome of lagging strand. The lagging strand happened because the replication happening in one direction and the elongation happening in the opposite direction. So due to this, the strands are, the pieces of strands are made and it will be repaired by ligase, etc. So it was discovered by two scientists, Okazaki brothers actually, and they got Nobel Prize. Wow, that's great. Some, in the old days, if you identify first time, you will get the credit. But now the science becomes so complicated, but still there are ways to identify new things to get prizes. Anyway, next one. Yeah.
PCR was developed.
PCR. Oh, very good. Today we achieved PCR, artificial DNA replication, just because of understanding this one. So one of the primary applications, PCR. Today we are able to do the PCR. Well done, right? So since you said PCR, I just want to tell one interesting story. Where is she? If she comes, then I will discuss. If not, she might visit the topic.
DNA application, there was a Tata box, right? Tata box also was there in DNA application.
Tata box. Yeah, that is promoter regions, this terminator regions. There are some. Now we are discussing about genes. That we said Tata box, that they are part of genes. We have a separate unit called genes. There we will study structure of gene. I will discuss that later. Okay. See, one problem we got was, can we able to replicate DNA outside the body? That is one big question. Can we able to replicate DNA outside the body? And what are the potential applications? All these things, whatever you say. We don't need a living organism to amplify the DNA. Just a machine can do the biological thing. So that is a very fascinating idea came to the scientist. Then there is one first generation scientist, his name is Sanger. So Sanger tried to understand the sequence of DNA in a very slowest method. Every day, after 4 hours of testing, lab experiment, he can identify one base pair. So he need to do 4 hours thing. And each gene has millions of base pairs. So sequencing the whole base pair with samples method is very, very slow. It will take 20 to 30 years just to understand a single gene. But by the way, very interestingly, Sander has identified 40,000 base pair Drosophila gene. He is very, very patient. See, every day he is doing 4-hour experiment just to know what is the next base pair. Is it AT or GC? It's very painful. Then, scientists need an approach of easily doing this amplification. Then, the problem they are encountering was, if you want, this is the DNA. If you want to attach a primer, the primer attachment happens around 60 degrees centigrade. Already we understood. Then, first of all, we need to denature the DNA. We need to remove the double bonds. We need to remove the helical structure. And phosphate bonds are so strong. Phosphate bonds are very strong. You know, DNA can be only be unbinded at 92 degrees centigrade, which is very, very, very high temperature. Just 8 degrees away from boiling temperature. Right? When we are trying to subject the DNA to 92 degrees centigrade, all proteins are denatured. Ligase, polymerase, SSBs, everything is destroyed. These proteins are destroyed. So, is there any possible way of replicating DNA with these temperature problems? I think PCR. So today we are saying PCR. This is the first generation scientist problem. Okay. So they are very confused how to resolve this problem. What molecules? Oh. So they have only one solution. They understood the problem. We need a polymerase enzyme that can able to withstand high temperatures like 92. Even at 92 degrees centigrade, this enzyme should not be destroyed. But there is no such thing. It's a damn protein and protein will be denatured. It's just like a small plastic material. You keep in hot water, plastic will be shrink. What to do? One day, there is a scientist, I forgot his name. Craig Venture and one more. Craig Venture, okay. So what I want to recheck his name. Anyway, Craig Venture, he got Nobel Prize. What happened? So he was driving his car along with his wife, okay, on a journey, and he's discussing same problem. Dear Melissa, I'm having this problem. We are all working in the lab. We are trying to isolate. We need to work on a polymerase enzyme. That enzyme should withstand high temperature at 92 degrees centigrade. But we are unable to identify that enzyme. What to do? Then she started smiling. Oh, is this a big problem to you? For this you are working for 20 years, research. They are going towards the Yellowstone in America. Yellowstone is a volcanic activity region. There are hot water springs. There are lakes with hot water. And clearly in that hot water there are some mosses, some algae. She said, dear Craig, that animals living, some mosses, some bacteria living in that hot water, that means hot water is at 100 degrees centigrade, boiling temperature, but still they are replicating, they are reproducing. That indicates they too have DNA replication enzymes, and those enzymes are working at high temperatures. Why can't you extract that polymerase, and why can't you use that polymerase for your experiment? And he was surprised of how she said this example. And he went to that pond and he isolated the bacteria. And that bacteria is called as Thermoaquatis. Thermal aqua creature. Thermoaquatis. So he extracted the enzyme from Thermoaquatis, and that's it. They solved the puzzle. They took the Taq polymerase, Thermoaquatis polymerase enzyme. They added to the human genome, and they started doing the temperature variation. And very, very interestingly, they got the extra strands. The DNA amplified successfully. And just like Rosalind Franklin, she deserves Nobel Prize, but he got and with his colleague Walker, they got Nobel Prize, but not to his wife. Even though she is a biochemist, by the way. Yeah, these things happen in the history. Yeah, that is how we got PCR, by the way. So PCR was discovered with a woman's help actually. But she didn't deserve the Nobel Prize, that's the problem. Yeah. So today PCR possible because we understood how application works. And the tag polymerase. Tag polymerase is like a golden, weapon for humanity. A simple thought from a layman talk. See, there are some wonders. Sometimes this happens. That's why by this I want to tell you what you learn. Rather than studying alone, group studies will have more ideas and you will learn better in group studies actually. Because you will get exchange of thoughts and that will make the concept very easier. And next one I need to say, rather than reading, try to teach to someone. Then you will learn a lot. Reading will have a slavery mindset of I need to study. But once you have the mindset of teaching the second person, you will expand your creativity to break down the topic to make it easier to the second person. In that procedure, you will learn it first. So that's why I haven't done Microbiology for past 5 years. But I'm teaching you confidently because I'm teaching you. That's the thing. That's why I'm keeping notes in my hand. I don't know. That's a teaching has its own significance. So next time you should teach to your friends and go with group studies. That will be very helpful. Yeah. So there is one question. Let me keep my PPT here. Let us learn collectively. Yeah. See. First question already completed. Replication of DNA. You already know all these things. We discussed all these things done. Ophthalmic fragments. You know what are ophthalmic fragments, right? So 5' to 3', 3' to 5', we have issues and the antiparallel strands will be discontinued synthesis. So yeah, this is all about ophthalmic. Next one. Role of polymerase, helicase, ligase and primase. Without saying, can you tell me what is the role of polymerase? Elongation of the DNA. Very good. What is the role of helicase? Unwinding of DNA. Very good. What is the role of ligase? Joining of the oxygen fragments. What is the role of primase? Yeah, to synthesize the primers. Right? Very good. So let us see what does the material states. Same things will be there. That's it. Okay. Primase synthesizes short RNAs. Very good. Ligase mix and ticks. That means the gaps of oxygen will be fulfilled. Helicase unwinds the DNA. Very good. Next question. These are genes. These genes I will discuss later. Okay, introns and exons. That I will discuss later. Future topic. Then, okay, these are genes. What is transcription? Do you remember what is transcription means? What is transcription? Translation. Do you remember what is transcription? Then what is the concept of central dogma of life? Yeah? Life moves in one direction of protein, the synthesis of proteins. How does the synthesis of protein happens? DNA to RNA, RNA to the protein. DNA to RNA is called as transcription. From RNA to protein is called as translation. So now you know what is transcription. Let us see. Transcription is a process of synthesizing RNA from DNA, making RNA from the DNA template. It catalyzed by RNA polymerase. Involve 3 main steps: initiation, elongation and termination. Obviously, RNA initiation, RNA elongation and RNA termination. Right? Yeah. So while fundamental process is conserved, transcription differs from prokaryotes and eukaryotes. Obviously, there is a difference between prokaryotic transcription from eukaryotic transcription. So initiation, what happens? So they are saying that there is a promoter region. I said that initiation, starting, promoter. So RNA polymerase binds to a promoter region of DNA and it goes, this -10 is base pairs, number of base pairs, 35 region. So Tata, he says it. So that is the promoter region. TAAT, it is the promoter region. Okay, Brin-Row box. Then -35 region. There are 2 regions. Okay, so that's it. Next one, eukaryotes has a Tata box. In eukaryotes we have Tata box, whereas in the prokaryotes we have a TAAT region. So now we understood what is initiation. Initiation facilitated by promoter gene. Promoter gene has TAT region. Simple. And there is a transcription bubble. Small, just like a replication fork, we have a transcription bubble. That's it. Then elongation. Common in both prokaryotes and eukaryote. So in elongation, definitely it will be elongated 5' to 3' by adding AUGC. Simple. You already know this one. So since it is a transcription, we are always saying RNA here. RNA polymerase, RNA elongation, RNA promoter region. Because it is a transcription. In transcription, what happening? DNA to RNA synthesis. Simple. Then last one. The third one is termination. So keep in mind, promoter region in eukaryotes is starter box, whereas termination. So termination sequence also we have. So termination is row dependent. I think there are some proteins for terminating. Row proteins, RHO. Okay? Row dependent protein termination as row independent protein termination. So row dependent and row independent. So there is a hairpin like structures that will do. If you know the structure of tRNA, there is a hairpin loop. So similarly, RNAs have some hairpin structures. So in eukaryotes, the terminating codon has a poly-A tail. Poly-A tail has a sequence called AAU-AA. Some things you need to be by heart, okay? So termination sequences and poly-A tail. Poly-A tail will terminate the transcription. This class is still 11th, right? Yeah. Then this is all about the transcription in prokaryote and eukaryote. Simple, it has three sequences: initiation, elongation, and termination. Initiation, Tata box, elongation, just by polymerase activity, then termination by poly-A tail and some sequences. Clear, right? Further things we'll discuss in the next lecture, okay? Attendance class. If you don't know all